So, hello everyone, and welcome to our webinar today on APC and large molecules. It's nice seeing so many of you here, or I can see that quite a few registered now, so thank you for joining us. For those who don't know me, my name is Jane Lucas, and I work as a marketing and event manager for Sophion. Today, I have the pleasure of introducing Juha Kammonen from River Laboratories, and together with Steve Trim from Venomtech. The two of them will give a presentation on the Venomtech and River Venom toxin library screen. So hopefully you will all enjoy this talk from the two guys. After Juha and Steve's presentation, our very own Kip Bodden, who's a research scientist at Sophion, will talk about large molecules in automated patch clamp. So feel free to ask questions in the chat forum. Use the panel on the right-hand side of the screen, and we will answer the questions after each presentation. So just shoot us with a question if you have any. If you'd like to learn more about APC and large molecules, we have added some applications report for you to download. So if you look next to the chat forum, there's a thing called handouts, and there you can download the free application reports that we have made on this subject. I think that while we wait for the last attendees to sign in, I would like to show you a short video about our latest family member. That's the QPatch Compact. Mads Korsgaard, who is the product manager for QPatch Compact, is also available for questions today if you have any after this short session. But anyway, some of you may have already seen it, but at least let me just put it live to you and show.
With QPatch Compact, you can conduct up to 8 experiments at the same time. Each experiment can be independent to investigate biophysical parameters, or they can be replicas to increase the statistical power of your experiments. In the Q plate where the cells are...
Sorry, I just restarted because it simply jumped in, so here we go again.
QPatch Compact is a semi-automated patch clamp system that makes patch clamping accessible to anyone. You do the pipetting. QPatch Compact does the rest. With QPatch Compact, you can conduct up to 8 experiments at the same time. Each experiment can be independent to investigate biophysical parameters, or they can be replicas to increase the statistical power of your experiments. In the Q plate where the cells are sealed and formed into whole cells, microfluidic flow channels and glass surfaces ensure good liquid handling. It is easy and ready to use when you are. Everything is explained on the screen and recorded for your documentation when making the analysis. Avoid the hassle and complexity of manual patch clamp setups. With QPatch Compact, you can focus on generating data. No hassle, just data. The data are stored on a separate hard drive inside the instrument, and there is automatically backup. You can analyze the data from anywhere without any extra software, and it is also possible to export it. You can place it where you want, though it is still so stable that you don't need to fear that anybody bumps into it and destroys your experiments. When you receive it, all you have to do is plug it in and turn it on. If you are interested in learning more about QPatch Compact, please check our website, sophion.com, or send an email to info@sophion.com. You can also visit our laboratories in Copenhagen, Denmark, Boston, USA, or Honjo, Waseda, Japan, to see and try it.
Okay, well, thank you for watching this short video. I hope you liked it. So I will now hand over to Kim, and he will give his presentation. So enjoy.
Thank you, Jane. I'll shortly introduce myself. I am a research scientist here at Sophion, and I am involved in some of the many wet lab R&D projects running here at Sophion, where large molecules is one of them. And on the screen, you can see that I'm showing a lot of molecules, so we can talk about this group and why it's getting more and more attention, not only by us at Sophion, but in general in the drug development community. And one way of classifying large molecules, or biologicals as some are referring to them, is via this line you see I made in green. So the small molecules drugs are the ones smaller than 1 kilodalton, and then on the other side you see the large molecules, hence the name. And here I show a few examples ranging in size from the small toxins or endogenous signal molecules, and then to the large antibodies. And from there's a lot of fragments like nanobodies, Fabs, and not bodies. The ones I show here are all protein-based. One can also include RNA, DNA, virus particles, and even living cells in the group of biologicals. And when you see on approved drugs, then the small molecules are dominant. More than 90% of approved drugs are small molecule drugs. However, the large molecules are rapidly rising in prominence and importance. And they've already gained dominance in the top-selling drugs worldwide, and this is why they gain so much attention. And one claim of fame is their mode of action, because they often achieve greater target specificity and potency than smaller drugs. But this also comes with a cost. They are expensive, they are scarce, and they can have a poorly reactivity. That's a fancy word of being sticky. And then in addition, they are sensitive to their environment. They have a three-dimensional structure, and this is key to their function. But this structure relies on weaker interactions and the covalent bonds that we're used to from the small molecules. And why automated patch clamp systems have been used characterizing small molecules for the last 20 years, then the use of APC systems in large molecule research has been more limited. And this is why we have set up some case studies to investigate whether special measures must be taken when working with this group, but also to highlight the functionality of the Qube and QPatch working with biologicals. And today I brought two studies. First, I would like to present a study on not bodies, which is a new generation of fusion proteins. And thereafter, I would like to talk about a study on snake neurotoxins and the possibility to neutralize the snake neurotoxin using recombinant antibodies. So first, I'm happy to share data on not bodies, this novel group of fusing proteins. And this was a collaboration between us at Sophion and Maxion Therapeutics. And please notice that it is Anish, Teresa, and Pedro that are the real heroes. They're the ones inventing and developing and manufacturing these molecules. But why not bodies? Because when developing ion channel drugs, it's a struggle to achieve potency and specificity, and especially with small molecule drugs. And you can get help if you look into nature's toolbox. Here you find animal venoms. They're containing toxins, which can modulate ion channels, like the pink toxin I show here on the slide. And a million years of evolution have made them potent, but often they lack specificity. On the other hand, antibodies have been employed to improve potency and specificity, but the design of antibodies targeting ion channels might be especially voltage-gated ion channels, because their extracellular domain is really small, so it's difficult to find an epitope. And then this epitope might only be available in a short time window when the channel is in a specific conformational state. So addressing these issues, the scientists at MaxGen have invented a new therapeutic class named notins, not bodies, where they're fusing the notin toxins with a antibody fab fragment, creating these not bodies that are then inheriting the blocking ability from the notin, but gaining multiple benefits from the antibody. And you see in the bottom that they have also fused full-length antibodies with the notin, creating not body IgGs. And in short, what can be achieved through not bodies is the ability to bind difficult ion channel targets. The larger size gives more degrees of freedom, more room for improvement, and then they have the enhanced in vivo properties of the antibody. And then in addition, they hold the potential for tissue selectivity via a specific binding. And to underscore this, I brought, there you go, an example of bispecificity. And on the upper graph, you see a parental not body. It's inheriting the toxins target specificity, but from there, 3 not bodies were engineered, and they are then recognizing both the toxin target, but also other targets of interest. And in this way, you can target both the ion channel, but the second target could then be organ specific or tumor specific or tissue specific. But it is not sufficient only to bind to an antibody. You also need to have functional modulation of the ion channel. And to investigate this, the QPatch came into the picture, allowing us to run 48 experiments in parallel. And we set up an assay evaluating the modulation of Kv1.3 channels by a notin and 2 derived not body fabs and 3 not body IgGs. And here in blue, you see a full Kv1.3 current response to a step voltage to 80 millivolts, and then this response could be modulated by the not bodies. And in purple, you see the effect of an IgG not body, and this is then followed by a full block of the SHK toxin. I also brought an IT plot displaying the Kv1.3 peak current over time. And here you see the rather slow onkinetics, and it's a feature we often see with these large molecules, and hence they need a relatively long exposure time. With this setup, we could perform a high throughput evaluation of the not bodies, evaluating the concentration response relationship of the molecules. And here I'm starting with the parental notin, and you can see it's extremely potent. And looking at the IgG not body from before, you see that some of this potency is lost when it's fused. But the IC50 value is still in the lower nanomolar range, and that is quite impressive. And furthermore, the IC50s for all 5 not bodies could be determined, and we could demonstrate a functional Kv1.3 channel modulation from all 3 not body, all 5 not bodies. And again, despite all the engineering work that these not bodies have been through, they still display clinical relevant IC50 values. Lastly, in this study, we reproduce all the data on the Qube 384 system, showing that it's feasible to run high throughput evaluation of biologicals on this system as well. And with this study, we can also demonstrate a reproducibility across our platforms. So in short, to summarize, then the not bodies are novel, is a novel molecule format harvesting the benefits of both antibodies and from the toxins. And the QPatch 2 and the Qube are suitable for characterizing these peptides, but also the NOT body function. I will then shift to another study. And here we evaluated snake neurotoxins and the neutralizing potential of recombinant antibodies. And this was a fruitful collaboration with Iontez and also the Tropical Pharmacology Lab at the Technical University of Denmark. And they did the antibody discovery and development. So, and while looking at snake venoms, and it's because snake bites is a highly neglected tropical disease, and almost 3 million people every year is being bitten by a snake, and it's causing hundreds of thousands of deaths and a lot of amputations. And currently the only treatment is animal-derived serums. And here you have to imagine that the snake venom is being injected into an animal, and then the serum is being harvested from this animal, and then it's being injected into the poor human that has been bitten by a snake already. So to aid the snake victims, then a novel type of antidote is currently under development, and it's based on recombinant human antibodies, and they are engineered to bind these neurotoxins and thereby neutralizing the effect of the toxins. And to evaluate this, we patched a human-derived cell line, and this cell line is endogenously expressing the nicotinic cholinergic receptors. And this receptor is the target of one of the most medically relevant group of snake toxins, the alpha neurotoxins. And with this setup, we're then able to study the neutralizing effects of different antibodies. So again, employing the QPatch, we made an acetylcholine dose response experiment, and you see the raw traces of the acetylcholine receptor response to increasing concentration of acetylcholine. And this we did to find an appropriate concentration giving 80% activation of the receptors. And this concentration could then be used to evaluate alpha-cobra toxin. This is a toxin from a cobra. And again, we could make a dose response curve, finding the IC50 value which we used in the following experiments. And when looking at the antibodies, they could indeed neutralize this current inhibiting effect of the toxins. First, you see a raw trace where you see the acetylcholine response and how it is being inhibited by the alpha-cobra toxin. But this inhibition can, in the second window, you see how this inhibition is cancelled by the neutralizing antibodies. And to determine whether a range of different IgGs could neutralize the inhibiting effect of alpha-cobra toxin, we tested 7 different IgGs, and we found that IC50s for these IgGs, and in this way we are able to rank them which IgGs are has the largest neutralizing potential. I can also reveal that the top candidate has now been tested in mice, and it's some really promising results we see from these mice. But when you look at the venom profile from the snakes, then each snake species is, the venom profile consists of several dozens of different toxins, and then when you're being bitten by a snake, you rarely recognize what species that bit you, or you often don't see the snake at all. So therefore, it's also highly desired to have cross-reactivity of these antibodies. And for this, we use the Qube to evaluate. The Qube is our system that can run 384 experiments in parallel. And here we tested 6 neurotoxins. You see them on top of the plate view. They were tested column-wise, and they were tested against 15 different antibodies, and those you see row-wise. And then this plate view, this is one experiment from one Q-chip, and that represents a heat map showing the neutralization ability of the antibodies, with red indicating that they have a high neutralizing potential, where the entire acetylcholine response is restored, whereas the blue, then you don't see any effect of the antibody and the toxin are blocking the receptors. And here you see that, for example, these two, these IgGs are having a highly neutralizing effect on one toxin, but only this one toxin, whereas there's other IgGs, like the ones down here, that they are having a more broad neutralizing capacity, so they are neutralizing several toxins. So this demonstrates that it can be achieved to reach cross-reactivity. Yeah, and then here in the end, I would like to summarize the two studies. So both the engineered NOT-bodies could block the Kv1.3 ion channels, and the engineered human IgGs could neutralize venom-derived toxins. And both the potency of the NOT-bodies and the neutralizing effect of the IgGs could be distinguished using automated patch clamp. So with this study, we both show that the QPatch and the Qube are suitable for characterizing toxins and NOT-bodies and antibodies, but also we have gained a lot of experience working with these compounds. And if you would like to know more about this, then we are in these days publishing a row of application reports on the topic. These reports are often decorated with exotic animals. And beside these reports, I think they're also attached to this presentation. But additionally, on our webpage, we have a lot of information, and then you're also welcome to contact me directly. So thank you.
Thank you, Kim. That was a nice presentation. We have a question here from Sibo. The question is, for recording Kv1.3, why a longer protocol more than 300 milliseconds is not used for fully deactivation? So did you get that one?
Yeah, I think for fully deactivation, yeah, you could do that. We only looked at the peak current, so therefore it would be it wasn't necessary. And when you extend the protocol, then you also you need to wait longer time between the pulses. So in order to speed up the pulse frequency, then we made a short, or relatively short pulse.
Okay, I hope, Sibo, that this answers your question. Are there any more questions to Kim's presentation? So please bring them forward, write them in the chat forum. You can leave questions there as well during the next presentation, so we won't miss them out. So anyway, thank you, Kim. We will go on to Juha and to Steve. I think that Juha will be the first to present. So I will just load the presentation here for you to see, and take it away, Juha.
Let me rewind a little bit.
You got it?
To the start. Hope you can hear me. Yeah, well, Juha's doing that to do a quick introduction. So I'm Steve from Venomtech, and so founder and chief scientific officer. And I can just see now that Juha's got to the beginning, so I'll hand back to Juha to start things off.
Go ahead, Steve. You might as well finish your intro, and I'll go on after that.
Yeah, so my background is in drug discovery biology. 10 years at Pfizer, mostly looking at pain and neuroscience, and therefore a strong interest in ion channels. Before being made redundant and setting up Venomtech to provide large molecules for screening projects such as for ion channels. And this is a great collaboration we've had working with Charles River on some of our toxins.
And I'm Juha Kammonen. I'm nowadays the group leader of the ion channel group here at Charles River at Chesterford Research Park in the UK. I worked with Steve at Sandwich down at Pfizer, so this is a nice way of closing the loop in a way. Again, on ion channels and pain. So it's like a very dear topic to us, I suppose.
Yes.
So we're kind of presenting work that's actually been done by others. So at Charles River, the work here that was done primarily by Elaine, who's on the call. Hello. And also Stephen. And at Venomtech, it was largely done by Danielle. So we have to thank them for generating all this excellent data. So we'll kind of get into this, and Steve and I will be swapping between the presenter role every few slides. I think I'll take a note from Elaine's book here and warn people that there are pictures of spiders in case you don't like them. We'll try and give you a heads up if and when they're coming up, but yeah, they are there. So the purpose of this project was to essentially look at finding novelty for sodium channels. Why did we look at sodium channels? I recognize a lot of people on the call, so we don't need to delve into this too deeply, but sodium channels are implicated in a variety of diseases, pain, different kinds of neurological conditions, epilepsy, neuromuscular conditions as well. There's a wealth of evidence to suggest that they are attractive targets to modulate, to basically improve the condition for a lot of patients. For example, we have desperate need for non-opioid painkillers. So that's the rationale for looking at sodium channels and trying to find something novel to modulate their activity. For us, why choose the Qube? At Charles River, we use this instrument routinely for a lot of drug discovery projects. It's our workhorse. We generate a lot of data on it, both single point high throughput screening as well as profiling for fully integrated projects. And for us, what we wanted to try and evaluate here is how well can we plug in this type of venom fraction library into drug discovery. And I think hopefully we can convince you that has worked incredibly well. I'll pass over to Steve at this point to discuss further about the rationale for venoms.
Yeah, thanks, Juha. Obviously, forming a company called Venomtech, it's obvious that we have an interest in venoms. And it's nice that Kim has just recently presented some of the knot-ins and how they interact with sodium channels. But the reason to look into venoms for ion channels is that they are a very good mechanism for either interacting with your prey or predators, and evolution has honed this over millions of years. Blocking sodium channels, such like ENEC sodium channels in insects, causes immobility and therefore enables the spiders to catch their prey. And those channels have also then evolved into the diversity of sodium channels that we see in the human body, and through serendipity, some of those are involved in pain and motor function. And therefore, these peptides that evolved to kill and paralyze insects then have some pain-relevant usage. But also, the spiders have a need to defend against their own predators. These big, scary spiders, which will come on the next slide, are not the top apex predators. They get eaten by other things, and a lot of those things are mammals. So activating or delaying inactivation of sodium channels has been a key evolutionary mechanism to cause pain and disturbance to predators. So there's a strong evolutionary reason for looking at these peptides. They're expressed in the venom gland and wait there ready for use, which makes them inherently stable. And we've got a huge diversity of species and therefore chemical matter to work from. So what I want to go to now is talk through a bit about Venomtech in the introduction. So I'm going to actually talk through the project, how we actually selected the venoms and prepared the screening libraries that Charles River then screened. So as I said in the beginning, we're specialized in preparing venoms for drug discovery, and we solve the supply problem by having the animals on site. And we can then actually recoverably collect the venom, purify it, and prepare the libraries, and we control end-to-end the supply chain, therefore making this a valuable resource for drug discovery purposes. And as Juha said, we have a shared Pfizer background. It's really nice that about 6 years ago we moved back onto the old Pfizer site, which is now called Discovery Park down in Sandwich. So why 1.4 and 1.7? Well, 1.7 is the sort of one of the poster child targets for pain, and having a strong background in that area, it was keen for me to have an opportunity to use our new libraries to actually find novelty and more chemical matter that can help drug development for this really important target. 1.4 is something new to me, really, in the sort of prospect of this project, and we had some interesting data looking at the mechanisms of spastic muscle contractions from spider bites that led us down the road of maybe this could be a Nav1.4 mechanism. So that's why we looked at the neuromuscular Nav1.4 and then used the Nav1.7 more as the traditional as a sort of technical control. We knew we were going to find 1.7 activators and blockers, and then could verify the technology as well as the systems. So our standard process is designing our targeted venom discovery array, or the TVDA, and the targeting is key here, that we don't just put any old venoms in any old libraries. We're selecting the right species and making sure that therefore you get a high hit rate in those libraries. Collecting the venoms, fractionate them, put them into screening plates, and in this case the screening was done by Charles River, and then identify the hits. Because these are natural libraries, we don't actually know exactly what's in each venom, so we then do a process of mass spectrometry to actually identify the hits we found, and I'll talk through that as we get to it. So on the prediction phase, we've had a great introduction to ICK, or knot-ins, or inhibitor cysteine knots. Now, knot-ins is a really good word for these, because they're not all inhibitors, as will come through in the discussion and see. Our selection process works on the fact of having a large collection of theraphosids, or what people classically call tarantulas, available to us, and actually picking the right ones for the right targets. And in this case, you've got sort of 3 grey spiders, but they're very different. So one's African, one's Asian. The sort of top 2 are arboreals, a tree dwelling, and the bottom one is an American ground dwelling spider. And then venom collection. So what we've got posed here is actually the shed skin of one of our larger spiders. It's nearly about 25 centimeter diameter, just posed over a HPLC vial, showing the idea of venom collection. And in the design process, we take our own internal data, as well as obviously the published data and everything else that's available to us, to actually get an idea of the best venoms to put in this library that generates novelty, as well as having controls in there. The figure here is from a British Journal of Pain review I wrote with my wife back in 2013, and we got permission to use this figure for that purpose. And what it demonstrates really nicely is that venomous animals from a wide range of toxin clades interact with ion channels both pre and post synaptically in a variety of ways. So it's just a great example of the diversity of chemical matter we have for this particular project. And obviously for us, we're working down on 2 particular sodium channels. When you look at a molecular mechanism, here is a diagrammatic sodium channel here, showing the classical toxin binding sites. So tetrodotoxin, a main positive control used in many toxin assays, got a binding site sort of here, slightly inside that transmembrane domain. A lot of the spider toxins are beta and delta toxins, so they're actually binding in similar place to the scorpion toxins, which are these extracellular loops in domain 2 and domain 4. But also you've got a range of different pharmacologies, including pore blockers, as well as those binding and alternating the voltage activation kinetics. So with our selection process, the key thing for us is actually that chemical diversity. It is ultimately generating a chemical library, and therefore we need the right hits to be drug-like and also act on the targets that we're looking at. Picking spiders that we know cause spastic muscle contractions on envenomation gives us a good idea about Nav1.4 activation. And interestingly enough, most tarantula or theraphosid spider bites don't cause that much pain. There's a few select species, and that implies that the mechanical damage itself is not enough. There's something else going on. And these give us a clue about some 1.7 activation. Putting this together and understanding the taxonomical relationships for those that we have data for and those we don't, actually built us a library that had inbuilt novelty as well as known peptides. And I've just put a list there about the beta delta toxins acting on the voltage gate, and then more like pore blockers from iotas and muse that are direct acting on other parts of the channels. So venom collection. So for us, we have about 185 species, and probably just over a third of those are theraphosid or tarantulas. Now, this actually is a drop in the ocean. There is 930 or so species of tarantulas alone, and therefore we've got a huge diversity of venoms to potentially pick from. Optimizing the husbandry is key. What we find that actually certain tweaks from the standard husbandry protocol can help optimize venom yields, obviously very important for this process. And then we operate a process of recoverable milking. So the spiders are basically like our herd. We milk the venom from them, they go back into their enclosure. And we do this under anesthesia, principally for the safety of the operators, but it's also good for the spiders as well. And then we can then take this venom protein. This is a protein solution up to 300 or 400 milligrams per mil. So it's a very rich protein solution. Quantify it for its protein content and then actually standardize it to start building the array. Once we know how much protein is in the venom, we then can put the same amount of protein into our two-dimensional HPLC system. So we use the Ultimate 3000 by Thermo Fisher, and this allows us to do ion exchange or size exclusion followed by reverse phase in a single inline process, which makes it very efficient and reduces losses from doing separate dimensions. Everything at the stock level is done in 96 well tubes with the 2D barcodes on the bottom, and then reformatted into 384 for the plating at the end. And then these are in echo compatible plates, although echo dispensing was not used for this particular assay. It's a standard SBS format 384 well plate, lyophilized and heat sealed, which means then they can be shipped to ambient temperatures anywhere around the world. For this particular project, it didn't actually need to go that far, just up to Cambridge and Juha and the team, which is where I will now hand over for the assay part of the talk.
Okay, so we have some electrophysiologists in the crowd, so I'll just very briefly kind of go through our methods on the Qube. We used the Charles River, Nav1.4 and Nav1.7 cell lines. Those are available and work very nicely on automated patch clamp systems. Our external solution was the normal Sophion EC000. Internal solution is a slightly tweaked IC000, so we have a little bit more internal chloride. And then we had a full block solution, so we could normalize our data to that and use that as an additional quality control at the end of the experiments to verify our seals. For all the experiments, as Steve mentioned, we received the lyophilized fractions. We dissolved those in our EC000 solution with pluronic acid. So we found from previous experience with known toxins like TTX and ATX2 that having pluronic acid in the external solution helps keep the toxins active rather than them getting absorbed into the plastics. We used a final fraction concentration of 2 microgram per mil for both the single point screening for hit finding and then also as the top concentration for our concentration response phase. We ran all the experiments with Goldcrest, which is the latest version of the Sophion software. Used multi-hole Q-chips. We found those work very nicely with our sodium channel cell lines, and with those we were able to reach a sort of 90% success rate, which is really quite nice for automated patch clamp experiments. And that, in short, is a summary of the sort of protocols. We use two different experiment protocols. So for those of you who aren't familiar with Qube software, it's slightly different in the sense how you build the protocols. So the Qube, you use these sort of experiment blocks. We typically always use a couple of liquid periods of just adding our vehicle, our external buffer, letting the seals and current amplitude stabilize. And for these experiments, we use the adaptive voltage protocols. So in these, we have an online Boltzmann fit shown in the bottom left corner here, which basically measures for each well the V half of inactivation. That voltage is then fed into the subsequent voltage blocks, and you can see it in the sort of bottom middle in the pink zone, where we can say essentially for this period of the protocol, either use the determined B half of inactivation, or if that determination is outside acceptable limits, then it will use a fixed voltage. In the single point experiments for hit finding, we use the top protocol labeled 1, but we found that actually we observed some seal resistance decrease, and therefore changed to just a single compound application. But also this was to ensure that we had enough material, since we were using basically pre-provided assay plates. So a few current traces, can't avoid them in an EFIS talk. Just showing here basically either Nav1.7, Nav1.4 current traces, very nice quality all throughout, and also just touching a little bit on how we analyze the data. So you can see in both cases, in blue, our vehicle control, so that was just EC000 with Plavonic. In orange, we have our TTX controls that inhibit the currents in both cases. And in blue, we have our ATX2 controls for activation. And just showing here where ATX2 very obviously delays the inactivation of the sodium channels. So these activator data we measured using area under curve rather than peak amplitude. For inhibition, we just used the peak amplitude of the resting state. And as mentioned before, for both of these, we got 90% success rates across the experiments. And one thing to highlight, all the data shown here is basically just from pretty much one experiment. No repeats needed. It was very, very successful. So going into the data then, we tested 960 venom fractions in single point over 2 experimental days across both Nav1.4 and Nav1.7. In the graphs here on the left, we're showing the peak current amplitude, either for Nav1.4 or Nav1.7. In the middle, we have the area under curve, which we used for activators, and then just some example traces on the right-hand side. And what we're showing in the graphs is, as the solid line is basically the line of unity between the 2 experimental days, and the dashed lines are our 3 times standard deviation for the DMSO controls. And what I hope you can see from here is that pretty much all the data that was generated lies within that 3 times standard deviation of the DMSO control. So it was very robust and repeatable data, specifically for the inhibitors. We have in orange our inhibitor controls. It's a very tight cluster, and you can see that we're kind of starting to get some venom fractions hitting similar inhibition levels as tetrodotoxin, and then also some activators showing up. And in the middle, you can see the area under curve data for activators specifically, It's not as clean as the inhibitors, largely because we didn't have an in-well activator control, so we couldn't normalize to the maximal level of activation within each well, whereas for inhibition we can normalize to the full block. So from this, we just started to find some very interesting looking venom fractions. I'll show a few examples. So to begin with, we have a non-selective activator. So here we're showing for each graph our vehicle control in blue and the venom activity in pink in the current traces. And in the IT plots in each of them, the first liquid period shows our vehicle, and then the next two show the effect of the venom on the current, and then we finally have our full block. So I hope you can see in these examples that for both Nav1.4 and Nav1.7, this particular venom fraction clearly changes the inactivation profile in slightly different ways for each channel. So there's a bit of a difference in activity already here. On the next slide, we're showing a non-selective inhibitor. Again, in a similar kind of way, in the current traces, our vehicle control is in blue, the venom fraction is in pink. We're showing initially in the IT plots the vehicle control, and then you can see how the current just decreases over time for each example. So we're seeing very nice biological inhibition of the current. And then finally, let's also start to kind of get us interested with, seeing some partially selective inhibitor data as well. So in the bottom row, the Nav1.7 example traces show pretty much full inhibition of the current. You can see if you compare the end of the second venom fraction application to the full block period, the current's pretty much completely gone. Whereas in the top row for Nav1.4, we're either getting about 50% inhibition or 20% inhibition in the second experiment. So, we're already starting to see some channel selectivity from these data. Going into the concentration responses then, I just wanted to kind of demonstrate some of the control data and how robust the assays were. On the left-hand side, we have basically our control well data. So we either have our EC000 vehicle controls in terms of the resting peak percent inhibition, or we have our TTX control, which is obviously full inhibition, and then also ATX2. The data there is slightly more variable because, again, as I mentioned, those data are normalized within the well. On the right-hand side, we're showing also the TTX control concentration response, which was in all of the plates. And you can see how tight that is across 2 experimental days, and error bars are also very tight. Our IC50 is pretty close to the reported value for Nav1.7 of about 20 nanomolar. So we were very happy with this. Out of the 960 fractions, we identified 33 to take forward. 26 of those were confirmed in this concentration response phase as modulating these channels. So I've shown a few example inhibitors here, highlighting again J19 in the top left, which we already picked out in the single point screen. And you can see that we're getting approximately 20 nanogram per mil potency for Nav1.7, but closer to 1,000 nanogram per mil for Nav1.4. So already starting to get some selectivity just with the venom fraction. Similarly, we also saw activators. As Steve mentioned, that the NOTINs don't only inhibit, they can modulate the channel in other ways. So here we're showing examples where we're getting activation of either Nav1.7 and Nav1.4, as in the top left graph, or activation of Nav1.7 only, as in the middle and the top right graphs. So again, starting to see some different behaviors with the different venom fractions. So in total, we had 32 fractions that we confirmed. and there was a different response profile between the two channels. This was very interesting for us straight away. And at that point, I'll hand over to Steve to just talk a little bit more about the next step.
Yes, this handover is exactly how the project handed over. So we had a catch up with Charles River team where they presented the data showing which wells of the array were the interesting hits. So interesting also to point out that they were completely blinded to which venoms were on the plates. They just knew the venoms in the middle sections and controls down the outside edge. We then worked back from our stocks, which is already provided for the concentration response curves, and then got that list of the interesting toxins. We looked at the data and the biology, and for practical and project related reasons, took 10 forward for identification. And this process for us is very standard. We do 2 forms of mass spec ID, both intact mass and a trypsin chymotrypsin double digest. This generates protein sequence data as well as intact mass that we can then use both to interrogate the published databases to identify these toxins, to actually find out the chemical matter that we've been using to modulate these channels. Starting with the activators on the left, really interesting data because people don't seem to look that often for sodium channel activators, so our chance of finding novelty was obviously quite high. I-19 was a known toxin with published activity on other channels, and we can add 1.4 and 1.7 to this known toxin's activity, which is quite exciting. And then also, we found novel theraphotoxins that have not previously been described actually acting as activators, and this backs up some of the previous data that we used in the prediction side of things, so we're really pleased with what we found here. Focusing more on the inhibitors, we selected a collection of both Nav1.7 only, as well as dual inhibitors, finding out what's going on with these mechanisms. And exactly as we predicted, we found known theraphotoxins, novel toxins, and also some variants of toxins with slight sequence variation from those that were published. So we're really happy with the library of activity we found from just taking a third of the total hits through to mass spectrometry ID. And this gives us sort of a good rounded result to the project, and therefore showing the benefit of screening a novel venom library. Even when we only took a third of the hits through for ID, we still covered exactly what we needed out of the project, getting that novelty as well as some known toxins in there as well. So it was really exciting to actually be able to do this with the new platform. And so for us, it was sort of verifying the benefits of using venom peptide libraries that have been two-dimensionally fractionated and therefore quite clean peptides, because deconvolution of venoms can also be quite a challenge, where we've sort of done this up front. So there's the benefit of actually working with the venoms. And now, for the last time, hand back to Juha to talk about the benefits and opportunities of the Qube platform.
Yeah, so there's a few kind of observations I think that we've made, and also just a few comments. So what's the advantage of using automated patch clamp? I think for us, one of the key things was that we can see the data, we can see how it looks, and QC based on that. We can look at the current traces, we can look at the current versus time, seal versus time, capacitance, and make sure that the data we generate is the highest possible quality. The examples here, essentially, in the top, we have one venom fraction where the second repeat in the pilot screen failed, but because we could see the data, we could see the current traces, the high quality in the seals, and also the IT plot with that kind of biological modulation of the channel activity, we chose to take it forward because we were confident that it was actually truly active. And that was confirmed in our concentration responses. You can see the activity in the CRC on the right. Similarly, in the bottom row, we have an example where, based on the number of looking at the percentage inhibition, this this might have looked like an active, but based on the IT plot, that just looks pretty much like current rundown. So we didn't think it was really active. We took it forward nevertheless, just to verify this. And as can be seen in the concentration response curve in the bottom right, it was inactive. So this is a major benefit of automated patch clamp. We can look at the data. We can put in place automated filters for such things as well. So we don't necessarily have to look at the data, but still can judge the quality based on those parameters. One of the challenges that we observed in the first part of the project, as I mentioned, was when we applied the venom fractions, we observed a decrease in the seal resistance over time. When we switched over to the single application protocol, we didn't really see this anymore. But I'm not sure if it's as clear cut as saying it's because of large molecules. I'm not sure that that's the case, but it was an observation that we made in this particular project. Considerations for testing venoms and toxins on the cube, certainly in this particular project. I think one message here is the data was really very robust. So what the graphs here are showing, either for Nav1.4 or Nav1.7, is the effect of each venom fraction at 2 micrograms per mil, either in the single point screen or the top concentration from the concentration responses. And I hope you can see that the activity tracks really very well between the two phases, between two preps of the same venom fraction. Now, because of the amounts we used, we weren't always able to get a full concentration response curve. We didn't see a plateau of the activity. However, once the venom's been ID'd, it's completely possible to then get more material for testing at a higher concentration. So that's no different to any small molecule drug discovery project. One thing, just as a practical note, because of the way that we receive the plates, it's best to try and use each plate for as many experiments as possible once it's diluted. But again, that's a minor practical consideration. I think Steve can comment as well. This is not really anything that will hamper a project in any way. So finally, in summary, what we set out to show with this collaboration project was how we could use these venom fractions in a drug discovery project, pretty much in a plug-and-play fashion. That's exactly what happened. For us, the key thing was that Steve and Venomtech were able to rationally select a library based on their information on how these spiders in this case behaved. We received the plates, tested them in the same way as any small molecule compound plates, and were able to find novelty and activity. So very quickly, we've been able to show that looking at venoms and venom fractions and toxins, we can find activity that perhaps has not been published previously. So for us, it's been a great success. I don't know, Steve, if you want to say anything before we do the acknowledgements.
I think it's a great place to move on to the acknowledgements. Yeah, we also saw it as a great success and some fascinating new data. So we're very pleased.
Yeah. So as I mentioned earlier, from the Charles River side, the kudos and credit goes to Elaine. She did a lot of this work with Stephen. I'm just the person who will hopefully get some emails on the back of this. And
yeah, on the Venomtech side, I was involved in the design process. Danielle, our senior scientist, did most of the HPLC and library preparation after Stuart had collected the venom, and Paul's our CEO that keeps me in check. And yeah, so like Juha said, I am the contact for the venom side of things. If you have any follow-up questions, that'd be great.
And I think as a final note, obviously this is an official collaboration between our two companies. So if you are interested in this type of project, then please get in touch. I think we'll finish there, Jane.
Yeah.
Thank you very much, both of you. That was very interesting. We do have a few questions here. I can see that Mads put a question saying here, let's see, Juha, the slow-acting inhibitors, what is your mechanistic explanation to that type of kinetic?
It's not necessarily just for me, this one, to be honest. I think Steve also needs to feed in here. I would imagine it's to do with where those act. As Steve mentioned, a lot of these may have different binding sites. They may be pore blockers, they may be voltage sensor domain modulators, or work in different places across the channel, and therefore they will modulate the behavior of the channel in different ways. If you have a pore blocker that just goes straight in and sits in the pore, that will very quickly change the activity of the channel. If you have something that binds perhaps closer to the voltage sensor domain and requires multiple openings of the channel, and that the channel has to cycle before the toxin can have its effect, then that could explain the slower onset of inhibition.
Yeah, I think it's also one of the benefits of using a larger molecule is they've got a larger interaction surface. So where the small molecules struggle with selectivity, the large molecules benefit because there's a larger interaction surface. And like Juha said, I think there's a lot of it's a sort of biophysics there of actually channel cycling and actually getting the voltage sensor domain accessible to this larger protein to act on it. But it is something we quite commonly see with the larger peptide inhibitors. I'm afraid I don't have an absolute, it's definitely this, but there is a lot of supposition around the kinetics of the larger molecules getting access to those voltage sensing domains, which seems to be quite key in the slow onset of this action.
Yeah, excellent. And Clint has a couple of questions here on the state dependent. Do you find rest preferring and inact preferring inhibition? If so, do you have binding site data? Would be interesting to see which domain sites give the state dependent MOA differences.
Yeah, so I've answered a little bit on the mechanism of action work there, but with toxins as a whole, so the diversity is such that there are both state dependent and state independent venoms, what we know of so far. With regards to which method of inhibition they're preferring, I'm afraid I'm not completely sure on that this time. I could certainly look into it. But it is interesting that there's a range of binding sites that I showed in that earlier presentation. And these molecules have, by serendipity, it's how obviously natural selection works. Something starts to modulate a channel, and then there's a selection pressure exerted to enhance that effect if it is beneficial for the organism's survival, which is a very different approach to organic chemistry. So there's a range of different actions, and what we know is that there's a large amount of biology here that we don't understand, and that's the exciting thing of looking into the novelties. So I wouldn't be surprised if new binding sites, new interaction mechanisms of action are discovered as we start to screen more and more venoms for these sorts of actions. We've also got the interesting convergent evolution. Juha mentioned ATX2, which is a cysteine knot peptide from sea anemones that has evolved to work on sodium channels to discoordinate prey, basically, or also potentially cause pain. So I think there's a lot more that as we start looking deeper with better tools, we will start finding more and start uncovering these new binding sites we hopefully can then use therapeutically.
Absolutely. I guess, Clint, at the moment we don't know necessarily the binding sites for all of these. That kind of work is ongoing, and Steve and I were laughing because just today I spotted a publication on PNAS on exactly the same kind of thing from the spider showing activity on sodium channels. So it's very opportune timing, and they showed some structure in there. So I think that work is ongoing. For these particular venom fractions and toxins, we don't have that information yet. Would love to follow up on that at some point. And in terms of the state-dependent data, it's in the data set. As I mentioned, we use that kind of 2-pulse protocol with the V-half of inactivation set for the inactivated state test pulse. So that exists. And we can mine that to have a look at what things look like with this particular set of venom fractions, if we saw anything like that.
Okay, thank you. And there's a couple of questions more. One additional question here from Clint. Do you find loss of seal resistance in empty hex cells, for instance, without now channels? Toxins can affect other targets, transporters, etc. Indicates challenges of using toxins as tools or therapeutics.
So we don't necessarily really observe that. As I kind of mentioned, I'm not sure if the seal resistance loss was as clear cut as being due to the venom fractions or being due to the double addition protocol. We know from experience that these cell lines and a lot of our hex cell lines cope absolutely fine with 2 additions of small molecules. In the concentration response phase, where we had the 1 venom fraction application only, the seals were absolutely stable. So it doesn't necessarily indicate that the seal loss was due to the venom fractions either. But yeah, in short, I don't think we see that kind of behavior with empty hex cells, if that makes sense.
And
Okay.
on the toxins acting on other targets, it's like a lot of drugs. There is other off-target effects, and the key thing here is that to start to do that sort of research to understand how these toxins are acting. Some have really exquisite selectivity, and others are broad spectrum toxins. So there's definitely a range of selectivities within the toxins.
Okay, and just a final question here. I think Mads puts this question here. Is it a large molecule culture thing to use weight / volume instead of molar concentration?
So I can pick that one up, and there's a very simple reason for this, and that is at the point we do the screen, we don't actually know the molecular weights of the toxins. So we can't look at a molar concentration. So we know the mass of protein, so we everything's weight per volume. Then when we get the mass spec data and have the molecular mass of the ligands, we can then go back and turn them into molar concentrations afterwards.
And I can see that Mark puts a comment here. It says, but even empty hex cells still express endogenous now and other channels, so it'll be hard to separate membrane destruction from ion channel effects. So yeah, that was just a comment.
Yeah.
Okay, so anyone has some more questions, or otherwise I would say thank you.
I have a comment from the person who did all the work. Thank you. This was done in
Good one.
X do not come into this. Thank you.
Teehee says Matt, yeah. Okay, so if we don't have any further questions, I think that we would thank the presenters. Thank you, Juha. Thank you, Steve. And thank you for everyone joining us here. And thank you, Kim, of course, for your talk. So it was nice seeing so many of you here. If you or one of your colleagues missed the webinar today, it will be recorded and we will share it at some time. So stay put. So thank you very much.